The 20 amino acids can actually be grouped according to the properties of their side chains or R groups. So this property is mainly if they are polar or nonpolar, if they are positively charged or negatively charged. For the nonpolar amino acids, these are amino acids whose side chains are mainly composed of carbon chains. So we have here alanine, whose R group is CH3. We have valine, leucine, isoleucine, and methionine. So methionine is one of those amino acids with sulfur in its side chain. Now, glycine is part of the nonpolar aliphatic amino acid, but compared to these other amino acids, glycine is relatively polar compared to them. We also take note of this very unique amino acid called proline. So in proline, its side chain is connected to the amino group. So this is not an amino group, but it's called an imino group because of its two bonds to two separate carbon atoms. So it's the only amino acid in our list of 20 common amino acids that has an amino group. And this structure of proline has actually a very significant consequence in the structure of proteins. So you will learn about that more when you discuss proteins. Next, we have the aromatic amino acids. These are amino acids with aromatic side groups or R groups. We have phenylalanine, tyrosine, and tryptophan. These amino acids, because of their aromaticity, actually display absorbance. And if you measure the absorbance of your proteins, you will be able to get some value. There is absorbance, and that absorbance is due to the presence of these amino acids in our proteins. So among these three, tryptophan has the highest extinction coefficient. It means that it has the greatest tendency to absorb light at around 280 nanometers, followed by tyrosine, and lastly, phenylalanine. So in average, we measure the absorbance of proteins at 280 nanometers. And we can actually use that absorbance value to estimate the concentration of proteins in an aqueous sample. Next, we have polar amino acids. Now, this polarity is not because of any charges. So they are polar uncharged side chains. So under this group, we have serine, threonine, cysteine, asparagine, and glutamine. If we observe their side chains, we see polar functional groups such as the alcohol in serine and threonine. We also have thiol in cysteine, and in asparagine and glutamine, we see here an amide functional group. So these are all polar functional groups. Worthy of mention among these is cysteine, because cysteine can form disulfide bonds. So if you have two cysteine molecules, these can go oxidation, and you have two sulfur atoms bonded to each other, in the process releasing two protons and two electrons. And again, just like proline, this formation of the disulfide bond leads to a significant consequence in the structure of your proteins. Next, we have the positively charged R groups. So these are also polar, but the polarity of the amino acid is due to the charge of the side chains. So we have lysine. We already saw lysine a while ago. We have arginine and we have histidine. So the positive charge is due to the nitrogens in the side chain. So the lone pairs of the nitrogen can actually accept a proton, and this gives the positive charge. In histidine, one of the nitrogens in the cyclic functional group is able to accept a proton. So in particular, this is the nitrogen that can accept a proton. So the side chain of histidine can also be positively charged. And we also have the negatively charged side chains. We only have two in this group, aspartate and glutamate. So their negative charge is due to the carboxylic acid functional group with the proton removed. Now, aside from these 20 common amino acids, we also have some uncommon amino acids that are worthy of mention, such as 4-hydroxyproline and 5-hydroxylysine. These two are derivatives of two common amino acids. So we have proline. So we just added an OH group in proline, in particular at carbon number 4. So this is carbon number 4, right? So this is carbon 1, 2, 3, 4. So we have OH at the carbon 4. And also 5-hydroxylysine has an OH group at carbon number 5. So this is carbon number 1, 2, 3, 4, 5. These two are found in cell walls of plants, and they are also components of collagen. Other uncommon amino acids include 6N-methyl-lysine. So this is another derivative of lysine. So it's derivatized at the amino group of the side chain. So instead of a hydrogen, it's a methyl group that is attached to the nitrogen. We also have gamma carboxyglutamate. So this is a derivative of glutamate. So in carbon number 4, this is supposed to be CH2 if it's glutamate. But instead of a hydrogen, there's another carboxylic acid functional group attached to it. So it's gamma because if you recall, this is our alpha carbon. So alpha, beta, gamma, gamma carboxyglutamate. Then we have desmosine. Desmosine is very unique because this is made from 4 lysine molecules. So these 4 lysine molecules are fused to each other. They form this central cyclic structure. And another unique amino acid is called selenocysteine. So instead of a sulfur or thiol group, what we see is a selenium hydrogen group. So it's a derivative of cysteine.